Hello everyone and welcome to today's DS lecture featuring my great colleague Birgitte Kallipolitis from BMB. Birgitte is professor in molecular microbiology and she's also head of the section of molecular microbiology at BMB. And she and we share a passion for understanding how genes are regulated, so how the genome is translated into RNA. That's what we think is one of the most coolest process in molecular biology, right? Yeah. But Birgitte has chosen to study this process in bacteria, and she has now for many years been working on understanding how bacteria is influenced by the environment and how that translates to different regulation of bacterial gene expression. And she's focusing specifically on pathogenic bacteria, and I guess we'll hear about this today. Birgitte obtained her Ph.D. Here at Odense University, it was called back then in '97. And then she moved on to a postdoc at Copenhagen University and was recruited back in 2001 to become assistant professor. And during that assistant professorship, she also did an exchange to Ireland And then she rose in the ranks and became a full professor in 2017, I think. And now she's heading the section of molecular microbiology. She's also serving at the Natural Science Research Council, so a very important position to have a representative. So Birgitte is a very important person at BMB, and we're really looking forward to hearing about your scientific work. Please.
Thank you very much, Susanne, for your kind introduction. Okay, I hope this works. Does it? I turned it on? All right, good. All right, well, thank you, Susanne, for introducing me. So, all right, okay, a bit too fast. All right, so as mentioned by Susanne, many years ago, I started out my own research group here at SDU, and we set out to answer these important research questions. First of all, we wanted to learn more about how bacterial pathogens, they cause disease. And secondly, we also wanted to figure out how to prevent infections caused by bacterial pathogens in the future. And along the way, we learned that by finding answers to question number one, well, this may actually help us to work out strategies for addressing question number two. All right, and that I'm going to tell you more about today. But first, a bit of background information. So question number two is actually more relevant than ever. We're running out of antibiotics, so we really need to figure out new ways to treat bacterial infections in the future. So this is just one out of many recent publications addressing this problem. Well, this publication describes the background and also the future that we're looking into. And the future looks pretty bad. So, as mentioned by these authors, drug-resistant infections could annually kill 10 million people worldwide by 2050. And they are mentioning this study over here. These are the numbers. So if we don't do something about it, by 2050, infections caused by antimicrobial resistant bacteria may once again become a major killer. So these authors, they also come up with some possible solutions. So they mention, for instance, we need new pharmaceutical targets to combat microbial virulence. And that's exactly what we are working on. All right, so the idea behind the strategy is that we want to target virulence and not viability. So pathogenic bacteria, they produce a number of virulence factors, such as toxins and adhesins, and these are all factors that are required for the pathogens to cause disease. So the antivirulence agents, they will prevent the bacteria from producing these virulence factors. So the idea underlying this antivirulence strategy is to design agents that block virulence rather than killing the bacteria, because that's how the traditional antibiotics, they work. They work by killing the bacteria or preventing them from growing. And this will force the bacteria to mutate or die. Okay, so relative to antibiotics, we believe that this antivirulence strategy, that it will delay the development of drug resistance. All right, so for these studies, we use Listeria as a model organism. So why should we study Listeria? Well, first of all, it's a foodborne human pathogen, and from time to time, it may cause serious foodborne outbreaks. And the worst ever outbreak seen so far in the world happened in 2018 in South Africa, caused by processed meats. There were more than 1,000 confirmed cases and 216 recorded deaths. The actual numbers were most likely much higher. We also see from time to time outbreaks in Denmark. The most well-known one dates back to 2014. It's called the rullepølse outbreak. We had 41 confirmed cases and 17 deaths. So this is rullepølse, cold cuts. So if you check out the numbers, then you will see that the case fatality rate is relatively high, usually between 20 and 30%. And that is even with early antibiotic treatment. And this makes Listeria to one of the most deadly foodborne pathogens that we know of. And it also shows that the current antibiotic treatment is simply not good enough. On top of that, Listeria is also a very popular bacterial model system. So we know that Listeria is capable of growing in soil, in food, and in the intestine of humans and animals, in blood, and so on. So it adapts really well to these conditions, and it also grows down at very low temperatures, down to 0 degrees. It can tolerate and adapt to low pH and high salt concentrations, and these are all conditions that are used traditionally for food preservation. So that makes Listeria to a serious problem when it comes to food safety. Furthermore, Listeria is also known to invade our cells, and it can replicate inside our cells, and it can also spread from cell to cell. So altogether, Listeria has become a popular model system for studying bacterial adaptations to stressful conditions, and also for studies on host pathogen interactions. Okay, so what happens during a Listeria infection? Well, first of all, it all starts out with someone eating contaminated food. Then Listeria will end up in the gut. And then within 30 days, Listeria may cross the intestinal barrier. Then it will enter the bloodstream, and it will be transported via the blood to the liver and spleen. And in susceptible individuals, it will multiply within these organs. It will then continue to spread via the bloodstream, and it may cross the blood-brain barrier and also the placental barrier in pregnant women. So in young and fresh and healthy individuals, you may just experience bad stomach, diarrhea, not very serious clinical signs. However, if you belong to the high-risk group, this infection may be fatal. And among the high-risk individuals, we have pregnant women and their newborns, and also adults aged 65 plus, the elderly generation, which is growing a lot in the Western world in these years. All right, as mentioned, Listeria has an intracellular lifestyle. That's actually why Listeria is so difficult to combat, but also makes it extremely fascinating for us to study in the lab. So Listeria expresses a number of surface-exposed proteins, including ional A and B, shown here. And Listeria will use these surface-exposed proteins to gain access to our cells. Then it will end up in this primary vacuole, and then Listeria will start producing a number of enzymes, and it will use these enzymes to break free, to liberate itself from the vacuole. Then it enters the host cytosol. In here, it will start to replicate and produce another virulence factor called ACT-A. And then Listeria will start to steal actin from our cells by using this ACT-A protein. It will build a tail from the host actin. And then it can use this tail to move and push itself into the neighboring cell. And then everything starts all over again. And that's how Listeria can spread from cell to cell. And it happens really fast. The generation time in here, in this compartment, is only 40 minutes. All right, so there is one single transcription factor which is absolutely essential for Listeria to turn on the expression of these virulence factor, and it's called PRFA. So the PRFA protein, it is a transcription regulator. And in its active form, it will bind to specific DNA sequences located in the promoter regions upstream from the virulence genes that are required for the intracellular lifestyle. PRFA has to be active in order to turn on the transcription. And if you make a mutant strain that lacks PRFA, then Listeria is completely harmless. And this, of course, makes PRFA to an obvious target for antivirulence therapy. So can we get rid of PRFA? Then Listeria will be harmless. All right, so this is a very busy slide. I'm not going to take you through all of it. This is just to illustrate that both the both the level and also the activity of PRFA is controlled by many different signals and regulators. So today I will just briefly mention to you a few of them. The high temperature, which has a positive effect on the level of PRFA. A tripeptide called GSH, which will activate or which will stimulate the activity of PRFA. And then finally, fatty acids, which have a negative effect on the activity of PRFA. All right, so let's take a look at temperature. So the expression of PRFA is controlled by temperature. In the 5' end of the messenger RNA out here, we see this inhibitory stem loop structure, and it works as an RNA thermosensor, which controls the expression of this virulence factor in response to temperature. So at low temperature, when Listeria is present outside of a host, this inhibitory stem loop structure will prevent the ribosome from gaining access to the Shine-Lugano sequence. This means that there will be no translation of PRFA, there will be no protein present, which makes sense because PRFA is not required when Listeria is outside the host. However, when Listeria then enters the host, it will sense higher temperature. This will lead to an opening of this inhibitory stem loop structure. Now the shiny organelle will become accessible, the ribosome will bind, and we see translation, and the protein will then be produced. So this all makes sense because PFA is needed for Listeria to turn on virulence gene expression when Listeria enters the host, body temperature 37 degrees. All right, so beautiful example for how a pathogen can adapt to signal from the host, in this case, temperature. Okay, so yeah, all right. So PFA can also be activated at the post-translational level, and this happens via a tripeptide called GSH, also known as glutathione. So GSH is present at high concentrations inside our cells. So when Listeria enters our cells, it will sense high concentrations of this tripeptide. Furthermore, Listeria will start to produce GSH itself, also when it enters our cells. So GSH will bind then directly to a tunnel site in PFA, and this will lead to a conformational change of PFA so that now it's fully active. As shown here, PFA is produced as inactive monomers. It will form active PFA dimers, but they are not active enough to actually turn on virulence gene expression. However, in the presence of GSH, it will bind PFA, become this fully active PFA dimer, which will be capable of binding to the PFA box and to recruit RNA polymerase. And there we have the virulence gene expression. Okay, so this is a very nice crystal structure of the PFA GSH DNA complex. It was made by some of our Swedish colleagues, and it shows very beautifully how this PFA dimer, what it looks like. Here you see that each monomer is, there's a GSH molecule within each, binding to each monomer. And then the dimer will bind very nicely to its DNA binding site. So this study kind of made the structural basis for the model that I showed you before. All right, so 5 years ago, our group showed that free fatty acids can actually inhibit the activity of PFA. What we did, Listeria will encounter free fatty acids in food, and especially also during the intestinal phase of infection. So we thought it could be relevant to study how does Listeria respond to free fatty acids. So we went to Niels Fauman's lab, and we got some free fatty acids that he had in his freezer. These were the 6 that he gave us. And then we went back to our lab, and then we added these free fatty acids to growing cultures of Listeria. And then we harvested Listeria cells, and we then tested the expression of the virulence genes. And to our big surprise, we saw that Listeria closes down the expression of virulence, of its virulence factors when we added these 4 free fatty acids. However, we saw no effect when we added these ones. So, but altogether, this first very simple experiment showed us that, well, some free fatty acids, some specific ones, may actually act as antivirulence compound by targeting PFA. So, of course, we had to look more into this, and we started to explore the mechanism underlying this effect. And to cut a long story short, we showed that PFA actually prevent PFA, that free fatty acids actually prevent PFA from binding to DNA. So this model shows or illustrates what we think is going on. So the free fatty acids, either they will prevent the dimerization step, of of the PFA monomers going into becoming active dimers. So either that step will be inhibited by the free fatty acids, or alternatively, they will somehow prevent the active dimers from changing its conformation into its fully active form. And in the next coming years, we will be working to figure out more about how the free fatty acids prevent PFA from binding to DNA. All right, so in parallel with this, we did another observation that actually made me quite sleepless for a while, because it turned out that some of these free fatty acids that shows an antivirulence activity at low doses, well, they also happen to have an antimicrobial activity at high doses, meaning that at high doses they inhibit the growth of Listeria. So that could actually be a potential threat against our antivirulence strategy, because potentially Listeria could develop resistance towards the antimicrobial activity. And what would that lead us with respect to the antivirulence activity? So we had to address two important questions. First of all, does Listeria develop resistance towards antimicrobial activity of free fatty acids? They probably do, because that's how bacteria act. They will always find a way to become tolerant and or resistant. And if so, does Listeria remain sensitive to the antivirulence activity of these free fatty acids? If not, we could just discard our antivirulence strategy. So we really had to test this and to figure out what was going on. We set out to do two different types of experiments. A Ph.D. student in our group, Rikke Thomasen, she tried to isolate free fatty acid tolerant strain and to analyze their response. And other members of the group started out to explore the general protection strategy of Listeria against these antimicrobial free fatty acids. So today I will just tell you a bit about Rikke's work. So Rikke, she simply added the high concentrations of free fatty acids to growing cultures of Listeria. And there they were. They showed up, the tolerant strains. She then genome sequenced them. And what she discovered was that the majority of these tolerant strains, they carry mutations in a gene called LMO 1079. And 1079 encodes an enzyme that is essential for Gluconate degradation of the waltikoic acids, WTAs. So, and they are carbohydrate-based structures that are linked to the bacterial cell wall. And the decoration of the WTAs means a lot to how Listeria responds to different types of agents. For instance, the decoration plays an important role in the response of Listeria to phage attacks. And also they play a role when it comes to biofilm formation and also to virulence. So Rikke's data suggested to us that the Gluconate decoration is also important for how Listeria responds to free fatty acids. All right, so she did a number of experiments to characterize these strains further. For instance, she did a fluorescent microscopy study, shown over here, to demonstrate that yes, indeed, her mutant strain does not contain Gluconate on the WTAs, and in contrast, the wild-type strain does, shown here. She also showed that she also did a hydrophobicity assay, and she showed that the tolerant strains, they have a more hydrophilic surface relative to the wild type. And finally, she did some experiments where she studied the binding of free fatty acids to the tolerant cells. And she showed, as shown over here, that the free fatty acids, they interact less efficiently with the surface of the tolerant strains relative to the wild type. So altogether, these experiments and more, well, Regis came up with this model to explain what happens. So yes, Listeria will develop tolerance towards the free fatty acids, and they will do so by simply getting rid of the gluconate on the WTAs. And that will then make the surface more hydrophilic, and then the free fatty acids will be repulsed from these tolerant cells. And that's what you see over here. Okay, so her final and very important experiment that we were waiting to see was whether these tolerant strains, do they then still respond to the antivirulence activity of the free fatty acids? And the short question, or the short answer is yes. So then I could sleep happily again at night. So what she did, this is just one out of many different types of experiments that she did, so this one is just to illustrate one example. So here she has grown the parental strain, which is of course sensitive towards the free fatty acids, and she compared it with the 3 fatty acid tolerant mutant strains. So she then split these cultures, and to one half she added an antimicrobial free fatty acid that also has an anti-virulence signaling activity. And what she could see then was that the expression of this PFA virulence, or this PFA regulator virulence gene, the expression of this gene is dramatically reduced after 30 minutes of exposure to the fatty acid. And we see exactly the same picture for the wild type as for the free fatty acid tolerant mutant strains. So that proved that although these isolated strains, yes, they were tolerant towards the antimicrobial effect, but they still respond to the antivirulence activity of the free fatty acids. And that means that our antivirulence strategy is still valid and it still works, and so we can continue to investigate this strategy. Okay, so this leads me to the conclusions. So as I've shown you here today, the free fatty acids, they may act as antivirulence compounds, and they do so in Listeria by preventing the key virulence regulator PFA from binding to DNA. However, Listeria may develop resistance against some of these free fatty acids, the ones that also have an antimicrobial activity, and they do so by getting rid of the gluconate on the WTAs. However, the good news are that we can still use the antivirulence activity of the free fatty acids to target these tolerant strains if they occur. So what are we going to do next? Well, we are right now in the lab, we are doing a number of studies with outbreak strains. So the results that I showed you so far, they were done with laboratory strains. But of course, we have to know whether the antivirulence strategy, it will work against the outbreak strains. So we have obtained some from Statens Serum Institute. And so far, the results look really, really promising. So that's great. We're also doing infection experiments with these outbreak strains to study the effect of free fatty acids on the infection process. Then we have started a collaboration with our Swedish colleagues, and we will hopefully end up with a co-crystal structure of PFA and together with the free fatty acid. We will also we're also setting up a genetic screen to identify where the free fatty acids will bind in PFA. And together, these two investigations should give us structural information on where the fatty acids bind in PFA, and this can also then be used as a structural framework for designing new drugs that could inhibit the activity of a transcription regulator such as PFA. And then we are continue to explore the general protection strategy of this pathogen against free fatty acids. Finally, as part of our future plans, we really hope to be able to do some infection studies in a mouse model with aging gut microbiota. And this experimental setup is part of an EU grant that we just submitted it yesterday, the 16th. And so fingers crossed, I really hope that we will get the funding so that we can do these experiments together with our colleagues in other EU countries. All right, so that leads me to my final slides. I would like to thank all the past and present members of the group that have contributed to this work. So these are the present members. In particular, I would like to thank Reggie Thomasen for, she did a lot of the work that I showed you here today. She's now writing up her Ph.D. thesis. She will hand in her thesis in less than 2 weeks, so she's really busy writing now. And also I would like to mention a former postdoc in our group, Patricia. So together, Patricia and Reggie, they did a lot of the important studies on free fatty acids and PFA. Also, I would like to thank Nils and his group for getting us started in the first place on working with these fatty acids, and we will continue to work together on this. And of course, also the groups from University of Copenhagen for their contributions on the structural studies. All right, so that's all from me. So thank you for your attention.
Thank you very much, Birgitte, for giving us insight into your exciting work. Have you patented this?
No, I have talked to Rio some time ago, years ago, about this. And because that the fatty acids, that they are just naturally present in the environment and they are part of our diet, they're just there, then they thought it would not be an option at this stage to actually patent this
not even
invention.
The concept,
yeah, I think when we are coming to maybe making starting to combine them and so on, then we are into something interesting. So so we should definitely be aware of that. So could be really nice if we could come up with a kind of a formulation, some specific fatty acids together that have a potent effect, and then we could patent that idea. Yeah,
definitely.
Yeah, so we are keeping, we are having it. We are paying attention to that. Yeah.
Yep. So I like to hear what are the questions I see in my hands.
Yeah, and that's a more practical question, Birgitte. How do you work with such bacteria there being so infectious?
Yeah. Well, we make sure to train our people really, really well, and then also to have a specialized laboratory. So we have a biosafety level 2 lab, which so everything is we are really taking very good care that, of course, the pathogens will stay inside this lab. We will not bring anything out from this lab. Everything is autoclaved and so on. And then the people that enter this laboratory and that gains access to actually work with these, they are trained really well and get personal instruction in how to handle these bacteria. It's of course we have to be really, really careful. So, but. No, we have not had any problems so far. So, yep. So, thanks to all the great taps and assistants, and training, in the lab.
Yeah, thank you for the very nice presentation. I really enjoyed it. I have a question. So, would you recommend now everybody eat a lot of the free acids if you feel like you were exposed to the pathogens? Or?
Yeah. So that's something that I hope to be able to say yes in the future, but we have to have the scientific evidence showing that, for instance, if you have like the right combination of free fatty acids, if you have those as part of your diet, and then you are more protected against certain foodborne pathogens, that would be really, really nice.
Are there any epidemiological studies that can help or resources?
Not yet, not yet. But that is something that we really hope to be able to get more information about in the future. Yeah, that's part of the mouse model experiment that I was suggesting there at the very end. So as part of that, we want to make different combinations of free fatty acids added to the diet, and then to see if the mice become protected against the infection. So it's worth trying. And if it doesn't work, if somehow the free fatty acids, they are metabolized or they are not stable or something like that, then we will still really like to know how these free fatty acids, they work on the regulator, and what the whole structural framework is, because then we can use it for designing alternative compounds that are maybe modified versions of the free fatty acids that are more like stable and will not be metabolized by the microbiota or by the host. But that's far into the future. But we already thought about that.
I have another very short question. You mentioned on one of the slides stressosome, and I'm just out of curiosity, what is this?
Which sorry?
In one of your slides, it was a cell with a lot of molecular process going on, and there was a stressosome, something?
Yeah.
I was just wondering what it is.
Yeah.
Out of curiosity.
Yeah. So the stressosome, that's a part of the, we don't know exactly what it is yet. So, but it's something that is under in investigation now. It's part of how Listeria will pick up stressful signals from the environment and then transduce that information to the transcription regulator called sigma B, which is a stress sigma factor that will turn on genes that are important for bacteria, in this case Listeria, to protect itself against stress conditions. So, yeah. Yep.
Yeah, thank you, Birgitte. It's a very nice talk. I'm also very much enjoyed. My question actually is at the very beginning is similar to the first question. Yeah. So probably we can provide the food or something containing the fatty acid. But my next question is, so you talk about your strategy is something about the antivirulence here. So, but what is the difference here between the antibacteria? Because I saw that if you provide, for example, enough amount of fatty acid, it will stop the transcription, the enzyme to the DNA. So will that also kill the bacteria in the end? If we kill the bacteria, then does it also antibacterial?
Yeah. So the point is that the same free fatty acid could actually have a dual effect, and meaning that one is that a specific fatty acid could both have an antibacterial effect, so that it would kill the bacteria at high concentrations, but then at the same time it will also have a signaling effect where it will prevent the bacteria from being pathogenic. So you could say actually that it's a good thing that one compound will have like a double effect on the bacterium. It will stop the bacteria from growing, and in the same time it will disarm the bacteria. So it will make the bacteria harmless, it will stop the production of toxins and so on, and in the same time, at higher concentrations, it will kill the bacteria. Yeah, so I don't know if that answers your question.
Yeah.
But it can
so
have
it's a
both
combination,
effects.
yeah.
Yes, it can have both effects. For some free fatty acids. For others, it's not like that, but let's save that for another time. Yeah.
And did you try the different type of fatty acid? The fatty acid is a huge number of different variations.
Yes. So in addition to the 6 that I showed you here, we did another study where we have tried a lot of different fatty acids, different lengths, different degrees of saturations, different degrees of, is it cis or trans double bonds and so on. And so we did a full study on that to get more information about what does it take from a fatty acid to be antimicrobial, to be antivirulent, and to be both, or not to be able to do any of it. So so we have a full picture of that. Yeah, thank you.
I actually think Kim was first, but now the mic is over there, so let's take a question.
Sorry about that. Thank you for a very interesting talk. So you've been mentioning low concentrations and high concentrations.
Yeah.
I guess, do you know what the intercellular concentration of these 3 fatty acids are?
Yeah.
Because I guess it's where it has to work.
Yeah, it fluctuates a lot. It depends on the cell type. It also depends, Listeria will go, so in the intestinal stage, we expect that the concentration will be pretty high because the fatty acids will be there as part of the diet. Then in the bloodstream, it will also be there. Inside our cells, it will also be there. But it actually depends also on how the diet of the person. Yeah, now you know a lot more about that than I do. It all depends also if so what does a person eat and how high or low the concentration of fatty acids will be in the bloodstream and in our cells and so on. But we believe that we can play around with those concentrations, and especially in the intestinal phase, that we have that we can play around with that and hopefully then affect the bacteria in that stage. Yep.
Now it's Kim.
Yeah.
Right, thank you. And fantastic work. I found really, really exciting. I was curious, so you partly touched on this already. So how big of a challenge do you think it will be that these fatty acids, they are probably buffered inside the hosting cells and maybe even metabolized?
Yeah.
You already touched on that, I guess. But maybe on the other hand, could that actually be exploited? Because you have a number of drugs that are known to modulate fatty acid metabolism inside cells. Could you actually tweak the cellular metabolism to produce these drugs?
That's such a nice question and suggestion, Kim. Yeah, that could be really, really nice sometime in the future to actually see if there's some kind of like a combination therapy, I guess, or combination treatment, so that if you can somehow, do something so that the host will have get higher concentration of the fatty acid that we want, and without harming the host, of course. But that would actually prevent the expression of virulence factors. That could be really, really nice. And that is something that would be really interesting to look into in the future. So that we don't just look at the bacteria, we don't just look at the fatty acids, we don't just look at the microbiota in the intestine, but also to look at what's going on inside the cells. What can we do to play around with the concentrations so that we can actually stop Listeria from replicating inside of a host cell by giving a drug you maybe already know, and that can be used for playing around with the fatty acid concentrations. Really, really nice, Kim. That's the future. I can feel it.
Yeah, thank you for a very interesting lecture, and I'd also say very pedagogical. So even as a historian, I was able to follow, or at least think I was able to follow your argument.
Thank you. I was hoping.
I had an argument with a question which is like outside the scope of your presentation, but thinking, so you describe this as a biological process, which means, of course, it's generic in a way. It should work the same way everywhere. But my question is, does the context somehow matters for these things? So, one context could be, for instance, how widespread is Listeria?
Yeah.
I guess you mentioned the cases where people die, but I guess there'll be a huge number of cases where people have like mild symptoms or mild cures. Also, you addressed that already, for instance, national diets.
Yes.
So it might be that it's much more dangerous in Denmark compared to, I don't know, Southern Europe or Japan. I mean, you have the same argument with aging. And also, I mean, another context which I think would be quite interesting is the political regulation of antibiotics, which is like hugely different if you look at in the world. I know there's some political scientists who will start to look at that. And I mean, of course, affecting people's health. So does it have some kind of interaction effects with the generic model you describe?
Well, this is just, I know it's like we're just looking into kind of a fishbowl and we're just looking at this. So, but of course we have some thoughts about, the applicability of this, and also more like in general terms. So can this be used for, first of all, as a microbiologist, can this be used also as a strategy to combat other bacteria? And the question is yes, because some of our colleagues that are working with Vibrio cholera, which cause cholera, and also Salmonella, you all know that one also. So some of our colleagues have shown that you can actually find similar effects of fatty acids on these bacteria. So I believe that it is a strategy that can be used for treating not only or preventing not only Listeria infections, but also infections caused by other bacteria. Okay, so that was one thing. The other thing is, okay, so as mentioned, we are a graying population. I mean, the Western world, we are becoming older and older. Or the number of people that are part of the population that are more than 65, that part or proportion will be growing also in the future. We know that. And that means that we have to do something to protect that population against Listeria. Otherwise, I think the numbers will go up a lot in the future. Also because Listeria is especially a problem when it comes to ready-to-eat meat, ready-to-eat food, and that is also growing. I mean, we want food really fast, pre-made, and that's where Listeria will pop up. So what I'm hoping, or what would be very nice to see in the future, would be, for instance, if we could have some recommendations from Sundhedsstyrelsen or whoever will set those regulations, recommending, based on our studies and our colleagues' studies, that, okay, if you're pregnant or if you belong to the elderly generation, we recommend that you add this to your diet to protect yourself against foodborne pathogens. That's cut very, very simple, but that would be a fantastic outcome of our study. Yeah. I hope that answers partly, yeah.
I love all these fantastic questions from different fields you're getting.
Yeah, that's great.
Would you allow me to ask a mechanistic
question?
Oh,
yeah. I'll see if I can
from a molecular biologist.
Yeah, sure.
So you showed in your model figure that these fatty acids, with these mutations, these resistant mutants, it's simply not getting absorbed or to a less degree.
Yep.
But apparently it's still getting sufficiently absorbed that it affects purify.
Exactly.
So would you care to speculate on that? Is that simply because that part of the mechanism is more sensitive, so the concentration of fatty acids can be lower?
Yeah.
Or is there like alternative ways fatty acids could be taken up?
Yeah, I think that I lean towards your first comment, that I think that so they are not like totally resistant. That means that if we that they can tolerate the higher concentrations of free fatty acids, but at some point they will also succumb. So that means that the free fatty acids, they will probably still be able to get access to the bacteria, although that the majority will be repelled. So I think it's a concentration matter.
So are there fatty acid transporters in the material?
No, they are taken up in a passive manner by kind of a flip-flop mechanism. Okay. That's how they enter the cells. And then
are you sure of that? Because that
no,
was also
we are not 100% sure. Yes.
That used to be the theory.
Yes.
How we thought that fatty acids were taken up in mammalian cells, but now we know it's actually their transporters.
It's a model.
Yeah.
And I admit that. So but that's what we know so far. We are lacking a lot of information on how bacteria respond to antimicrobial-free fatty acids. So it could be in the future that we will learn differently. But so far, that's the mechanism.
Excellent.
Thank you.
Any further questions? If not, let's thank Birgitte once more for a fantastic simulating.
Thank you.
And thank you all for coming. And please remember, every Wednesday there's a great DS lecture. And next week it will be Sebastian Mernil talking about his work. So put a note on your agenda.